Hello, everybody.
Hello.
Hello. I'm Fazalam. This is my colleague Danny Chan. We're both both have PhDs in microbiology, and we're both interested in microbiology. So we're gonna talk to you about papers. Um, this, So this week we're talking about pathogenesis and transmission of SARS-CoV-2 in golden hamsters. Uh, so,
Yeah,
uh,
if you were joined us last week, we looked at a whole bunch of different journal articles, and we thought this one seemed the most interesting. So today we're gonna dive into the figures one by one and talk about the data and our
yeah,
view on what they presented to us. Yeah,
yeah,
so I just want to say that they link to like a review, like where they've used these golden hamsters for all sorts of different viral infection models, right?
yeah.
This is like yeah, these hamsters are definitely definitely a model organism. Maybe not as popular as mice, but some popularity, you
Yeah.
know? Hmm.
The interesting thing about these golden hamsters is the H2 receptor. So if you've been tuning into previous episodes of Micro Journal Club, you'll know that this is the receptor that the SARS-CoV-2 uses to latch onto cells and get inside.
Yep.
And there's been a couple of studies that have looked at that found that the hamster version of this protein is quite similar to the human version, much more so
Right.
than the mouse. So in
Right.
previous models, they've managed to use that to successfully model infection of the original SARS original version.
Yes. Yeah.
Um,
They've infected these hamsters before with original SARS
yep,
and shown that it replicates, and the original SARS has the same ACE2 receptor. I think there are some differences between the spike proteins, but yeah, they use the same receptor. Um, and actually, I mean that feeds right into our first figure. I don't know if we want to say anything else about things that they did before. I mean, they've done like MERS um, is a different receptor, but they've used that before, right? To infect these hamsters. So I mean, these hamsters have been used around this research for some time. Yeah, they're well known in that way. And I guess that means they also have hamster reagents, right? That's the other important thing about model
yeah,
systems is that like, do you have the ways to well, besides the housing, but like, do you have like the antibodies,
yeah,
right,
exactly.
to stain certain like immune cells inside of a hamster, or like the hamster version of different proteins, so you can identify cell types, right? That all comes with the model, and we know mice have tons, and hamsters do too.
Yeah.
These hamsters do as well.
So, yeah, I guess the first slide we're looking at is just the basic infection of the of the hamsters. I know I'm gonna mix up between saying mice and hamsters because that's just my. I'm just gonna
Small rodents are all the same.
Yeah, small votes all look alike. Oh gosh, I'm gonna get cancelled now. um, yes. So the first actually figure looks at. So they infected the animals with the SARS-CoV-2, and they looked at the infection. So like,
Oh, actually, wait. I just want to say I forgot. In the extended figure one, they actually do show that alignment that you were mentioning.
oh yes, of course,
Um,
yeah. I did not
y
put that in my slides, unfortunately, but
oh,
I can
that's okay. I just thought that was
holding
actually
up the
that was a good
camera.
thing. Yeah. Yeah, but they did right. They do actually do a amino acid alignment against hamster, macaque, and mouse. ACE2.
Yeah,
And it's evident from the alignment that there are a lot more amino acid differences between the mouse one versus the macaque, hamster, and human.
yeah,
And that makes sense because we briefly talked about this in the previous episode, right? That the mouse model has to be jimmied up to make sure that infection can happen, right? You got to either replace the virus's ability to get into the cells with a different spike, or give the mice the right receptor.
yeah. I mean, we briefly looked at two papers on that, which were crazy, but this is actually does it naturally which is
Yep.
kind of good because that kind of evolutionary similarity could play another role between hamsters and humans. So,
Yeah,
uh,
yeah, absolutely. I mean, that's the whole model thing, right? Is that you're studying this thing. It's not the real human immune system,
mm,
but it has similarities, and we're hoping that those similarities are meaningful enough to speak towards, yeah, to actual us. Um,
yeah. Uh, so yeah.
Okay, yeah, so they infect the hamsters.
Yeah,
Or
so
like
they infect
how
the
do they do it?
hamsters through the nose. They like instilled a bit of like liquid through the nose, and then they sampled using nasal washes, which
Hmm.
is so I don't describe they don't describe in detail, but I do kind of recall when doing similar things in other animals. You just pipe water, liquid in and out,
Yep.
and
Yeah,
hopefully you
I
get a
think I read
sample.
that it was done under anesthesia.
Yeah,
So
it would
yeah.
have to be. You alive. You don't want to do that with an animal that can is conscious and can be really upset with it and also bite you and
Right.
or bite the whatever you're using to. It's it's much better to work with some animals under anesthesia just for
I mean, certainly when you're doing things that are so invasive, right? Or like in the sense that like you're sort of waterboarding
yeah invasive
the rodent.
and
Yeah.
also it's dangerous. Like if an animal suddenly like squirms out your grasp and runs around the SARS-CoV-2 around your lab, you're
That's
kind
right.
of
Oh yeah. Also remember these are
whoops.
yeah infected with SARS-CoV-2. Actually, like the variety that infects humans, right?
Yeah,
They have
exactly.
an isolate from some Hong Kong patient that they've grown up.
Yeah, and that yeah, so you do want to have them knocked out for not just for humane reasons, but also for the various inhumane reasons as well. So
Hmm.
that's not an excuse if you
Yep.
yeah. So being a psychopath is not an excuse for being cruel to animals. That's just something that need to establish because it's not scientific because that's not going to be a good excuse.
Yeah.
Yeah, and
And so, yeah, I think it's cool that they do the nasal pathway. So this is unlike when we saw with the macaque model, right,
oh
where
yeah,
they wanted infection however they could get it, right, with either because of the value of those animals or because like they're not sure, right, like
yeah,
exactly what will work. But here we're looking with animals that maybe they're a bit less one-to-one valuable as like the animals using those macaque studies, and they're going to end up talking about transmission, right, as something that you can study in this model. So maybe we want to see the infection come in a way that a way that's more realistic, right, to the human condition, right? We
yeah,
think that we're getting it through our nasal pharyngeal.
yeah. So yeah, they measured the actual viruses produced each day using well two techniques. There's either counting the viral RNAs that are present in a sample, and also checking whether there's any virus that can infect actual cells.
Yeah,
So that's
yeah. So we've definitely talked about the counting viral RNAs way, right, through
yeah,
RT-PCR before. Um, Although, yeah, well, we also last was it like 2 weeks ago we talked about like there's actually two ways, right, to count that viral RNA as well, right? It depends
yeah,
what marker you use can give you different signals.
yeah,
But for their corroborating secondary, they use probably a more relevant and also very functional way of counting viruses. They use plaque assays
yeah.
um, and
So
actually count the amount of viral particles.
yeah, so
Hmm.
plaque assays are great because they literally have proper cells, and if the virus is there and live and it infects them and kills them off, then you know that it's actually a working functional virus. Whereas
Right.
an
It does.
RNA, yeah.
Oh, go
No, no, no.
ahead.
I was about to say like with the RNA, you don't know whether it was functional or not, and that's
Yeah.
actually quite an important thing because so in this there's like the blue columns and the red columns. The red columns are the RNA, and the blue columns are the viral titers.
Yes.
And
Yeah, and you can see that. So in the lungs, they have 5 and 7 days, right, post-infection. And you can see that the viral RNAs are still present
yeah,
by the seventh day, but they can't find any plaques, or at least it's below the limit of detection.
yeah, and also they also mentioned showed the kidneys as well, where they didn't really detect any live virus, but they did detect some viral RNA, which kidneys are since they filter out all the blood. So if something's in the blood, will probably turn up in organs like the kidneys or the spleen. So it's
Hmm.
a kind of a way to check whether a virus has gone systemic, where it's gone big and gone all around
Yep.
the not the human the hamster body. Of all
Oh,
the
mice, hamster, humans, hamster.
we all look alike. We're all mammals.
Yeah, actually, it was interesting in the introduction for the paper. Like they cited a paper where they just looked at human tissues and the ACE2 expression in it, and
Yeah.
they were saying that you can find ACE2 mRNA. So like we make that mRNA in many of our cells actually, but then they actually went in and they sliced up tissue to see where it was, and they find it on a lot of different. it's not just in the lungs that you have this ACE2 receptor, right? It's in a lot of other
Yeah.
cells. And I mean, that when you think about if the virus could get into the blood, right, it then has access to all these different tissue types, right?
Yeah.
First, endothelium. And then if it's able to pass through that, right, maybe something's a bit leaky, or like maybe there is an inflammatory response, right, and it gets to move out, then it could also access other tissues, right, other organ functionalities.
Yeah, and that's an important consideration for pathogenesis, especially there has a story been developing about COVID attacking the blood vessels. So
Yeah, because of all the hematologic symptoms, right, that
yeah,
they're seeing. And so, this is also a feature, I think like several months ago, we couldn't say like models are trying to capture features of disease, right? Several months ago, when we were maybe talking about the macaque model, we couldn't talk about that feature of disease, right? Because we
yeah,
didn't really know, right?
exactly.
We just, not that many people would be infected, we didn't know. And now that we have all this human data, it helps to come back to the models and inform the way that we look at them, right? Because if this study was done like before we knew all of this, maybe they never even would have looked.
Yeah, exactly.
Right? That might not have been something that crossed their mind. Like there's this interplay between, like what are we trying to figure out and how are we going to measure the things in the model, right, to support that information. Yeah, so like we know that there potentially is an effect, so they would like to show us in the hamster model if that's the case, and we'll show plenty of histology
Yes.
of the different tissues that they looked at.
Oh yeah, we have got lots of histology to look at today. Because they did take samples of organs at days I think days 2, days 5, and days 7 to look at the progress of infection. So in different organs as well. So we're going to take a little tour through that. So the first set of slides is figure 1C and D, which are looking at the lungs 2 days post infection. And so side by side you see these two slides of a lung section, one stained with hematoxylin and eosin, which I think that stains nucleus, because I literally wrote on my paper research what H&E does, or remember, because I know like one of them does nucleus, and the other one does
nucleus. The eosin I think is for the nuclei.
Yeah.
And then I'm not sure what I'm not sure what the other one is.
But yeah, so
but what you end up seeing is dark. The purple is dark nuclei, and you can tell a lot just by seeing the borders of cells and the nuclei that are in them. I mean, we've seen this data before in the macaque model or whatever, right? Like when like immune cell infiltrate, right? All immune cells have a nucleus, and so when they're pouring into tissues, you'll see like a cluster, right, of these like. And also immune cells have a certain morphology, right? They'll look kind of lymphoid. They'll be like a blob, opposed to like a column or like neatly ordered.
Yeah. And I find like blood really stands out as well as like quite red in it as. So I think you can see some blood vessels in here as well. But indeed they actually stained for the virus to show where the virus is being infected.
Yeah.
And
so they're looking at Oh yeah, indeed.
Yeah.
Sorry, I thought you said indeed. I'm like, yes, indeed, they did. They did look for the virus. Oh, indeed.
I am too British sometimes. My mistake. That is something I would say.
Yeah, they're looking at a viral antigen, right? Because the cells that get infected are becoming viral factories. So on the outside of those cells, there will also be the antigen of this virus, right? Like the surface proteins of the virus are being expressed on the surface of these cells, and that's what we can see.
Yeah, and it's interesting because that's a bronchiole, I think, an airway. So you find that as you'd expect, the airway is the first part to get infected by the virus. Which
2 days post-infection, all of those cells have been exposed.
And then we look at 5 days post-infection, and you can see the like kind of there's a lot more infiltrates around so you can see very dark areas where there is that inflammatory infiltration.
So I think it's definitely visible like around the white spaces, right? Like so the white spaces are presumably like air pockets, right? Because the lungs full of these air pockets. And again, I think we may have talked about this on the macaque one already, and then seeing that purple cluster around it. Or I think even they don't have a zoomed in image in this way, but if you zoomed in closely, you should be able to see what that epithelial layer looks like that lines the airway, right? Like it should be like a thin layer of cells, and if there's infiltrate, right? It means that there'll be cells on both sides of that thin layer, right? And you're like, okay, well, something's happening here.
Yeah. And again, you've got like those like brown blotches, which is where the COVID proteins are showing up as well. So
Hmm.
so it kind of shows that these lungs are getting infected. The infection is getting worse. But on day,
Yes.
oh yeah, and also on day 5, they also, so this goes into the external like, so extended figure 2, uh,
Hmm.
2b, where they where they look at CD3 T cell infiltration. Uh,
Yeah. So
at
this is great. This is resolution that we haven't seen before, right? When we've looked
yeah,
at models.
yeah,
Because it's one thing to say, "Oh, I see these nuclei are just like
yeah,
getting into there." But like what are those cells exactly? Like
yeah.
they have molecular markers that we can use using these staining techniques to figure out what they are. So that's 2A. Oh
2b,
no, 2B.
yeah,
Because 2A they're actually looking at like a morphology of
yeah,
the lung cells themselves, right?
yeah.
That they look like they're
2a is
proliferating.
from day 7, so it's jumping ahead
Mm.
to the
Oh yeah,
happier. So that's why I've put things a bit out of order because I
I
was,
see. Yeah, you're helping us order it in the narrative of the paper. Understood.
yeah.
Not the
So
narrative of the figure.
this morning I was just scrambling, thinking, oh, how do I do this? I'll just do it in,
Yeah, yeah, that makes
yeah,
sense. Yeah, so
uh,
there's these brown foci, right? That's what we're looking at in B. These
yeah,
like brown lumps, and they look like they filled in the air spaces actually, right?
yeah.
Where there
You
could have been air spaces.
Yeah, I mean,
There's
it
now
even
these
seems that
brown foci.
some of the S spaces have like kind of like you see bubbles. They should be those bubbles should be like circular, but they've joined together almost as if parts of it have eroded away. So they
Yep.
so that could be evidence that there's been some damage to the lungs caused by this inflammatory infiltration.
Mm-hmm.
Um,
Mm-hmm.
but I mean, I'm going to qualify. I'm not a proper qualified histopathologist, so I could be getting this. I'm just looking at these squiggles and making my story around them. So I'm not,
Right. I mean, that's what they told us in the paper, right? That's
yeah,
the other thing, right? Like, yeah,
yeah.
you can only go so far in like how you can criticize the data yourself, right? How the data
Exactly.
come together, anyone can sort of tell that story, but it does take a lot of people to like really. It takes a certain type of trained person to look at a slide and say like, I know what this is.
Yeah.
Uh, and like for infectious model labs, right? We were both in
Yeah.
labs that did infectious models. People become acquainted to seeing their specific tissue type, right?
Yeah.
Healthy and sick, and what the feature is that you're looking for between the two, right? And
Right.
in a paper, you just say that comes out to one sentence, right? We see the infiltrate, right? We see evidence of disease,
Yeah.
right? But like on the working side of it, it's like, well, they've seen many different
Yeah.
instances of this, right? Every time they're seeing it.
No, that's
But
no. You keep going. I was about
oh
to
no, I
agree
was just
with
gonna
you.
say, but yeah, but it's great that they are showing us a little bit of that process, right? Instead of just saying we saw the evidence of it, now they're going to show us a slide where here is an example of what an infected looks like, and that positive staining that they're using to make the conclusion that there are immune infiltrates in these air spaces.
Yeah. Uh, that's brilliant. And 7 days, the lungs seem to have started to heal a lot more. So it so they have like a they mentioned they see type 2 uh, pneumocyte hyperplasia, which I did
Yeah.
look up. Which so pneumocytes, there are two flavors of them. One of them is the normal lining where the oxygen passes through, and the other one which produces lung secretions that also, and when the lung is damaged, they start dividing like mad. And so
Mm-hmm.
essentially what they're showing is these cells are dividing like mad, and it's the lungs healing essentially. So the worst of the infection is over, and the lung is starting to rebuild itself.
Mm.
And and I
And
think,
also evidence of something, right? Like
mm,
it's not just that the virus can go in, replicate a bunch of times, get cleared by the virus, but there's some lasting later effects that are coming
yeah,
down, right? That like that that means the lung has to go through a process. I mean, again,
yeah,
that's probably obvious, but being able to see it in a model paper is trying to identify what like does this if this correlates to something in humans, can we study that?
yeah,
Right? Is this a piece of data that could be useful down the line,
yeah.
or an endpoint, something like that, right? Something to measure.
And it's important to also get the right time scale of everything together to see
Yeah.
like when is the lung able to heal itself, or is
Yeah.
it and is healing? And so extended figure 2A delves into this more, where it like looks into more into towards the hyperplasia kind of those cells. Again,
Mhm.
this is a really complicated histograph that I'm fine because it almost looks like a magic eye picture to me. I'll be honest.
Yeah, I'm zooming into it to try to see the detail better. It just looks like a lot. Like I don't see what the features are when they're
Yeah,
saying hyperplasia. I mean, I assume what they're looking for is they're looking for some layer of cells that's getting thicker,
yeah.
right?
I mean,
um,
it could be because I'm not seeing air spaces around here, so I'm not sure. Like, I mean, if
mhm.
so, I'm not sure what part of the lung this is, or so I'm a bit at sea. I do see like one big blood vessel there, but in like the bottom left-hand corner. But I'm not
Yeah,
sure
yeah.
about the rest of it. But again,
We're gonna have to
I'm
trust the authors on this one,
yeah,
right? Like they
basically,
know what they're looking at, and they've tried to show us the evidence. So again, if someone else could look at this, they'd be able to make that judgment for
yeah.
themselves. But for us, we'll just say
So yeah,
we can see
histopathologists in the comments, please
it. Yeah, we need more
educate
histopathologists.
us. Yeah. Um, and the next part, they move to the nasal turbinates. So, nasal turbinates, they're like, so your nose is the bit in the front, but right at back in, like, goes inside your skull, the turbinates, which are these really folded up bits of, like, flesh that kind of do all the smelling and basic
Yeah,
sensing and humidify air before it goes into the lungs, so you don't dry out too much.
we have
Um,
them too.
Yep.
We
I
have them
mean,
too.
most animals have them, and the interesting thing I found is that because they actually like kind of back up against the brain because kind of evolutionarily the sense of smell has been very important
Mhm.
for animals. So a lot of them had backed up right against the olfactory bulb, and
Mm.
yeah,
Low latency connections.
yeah, exactly,
Shortest distance to fiber optic cable.
yeah. So I mean, and it's interesting here because they look at like again what COVID attacking the nasal turbinates at different times post infection, and then they
And this
go
was again, or I was gonna say, this is something that they had, like that idea of like the sense of smell. They had talked about that as a symptom, right? Like
yeah.
people have reported like some light sense loss of smell or whatever. So I mean, again, this is like what's focusing us on this area, both because it's the site that is going to get the path of the air, but also because it has some symptoms associated with it.
Yeah, completely. And they do actually go a bit deeper into this. Mostly, mostly in the extended figures, but I think that's quite worth looking at because they mostly do a lot of work on co-localizing, seeing whether is COVID doing anything specific to the nerves. So,
Hmm.
uh, so in extended figure 2C and D, they've got they've stained. So in 2C, they've stained a section of an infected hamster with an antibody that stains for COVID-2 that shows up red, and an antibody that shows up that stains the neurons brown. Well, stains a protein on the surface of neurons brown.
Yep.
Um, and like and in D is basically a control hamster that hasn't been infected, so you can kind of compare, see what a healthy a nasal turbinate would look like compared.
Hmm.
Um, so again,
Yeah,
these.
it's. Oh no, go ahead.
Oh no, no, I will let you talk finally.
Yeah, so it's interesting. Like, oh, it's hard to zoom in on these pictures. Maybe they have like a limited resolution and won't let me zoom directly into them. But it's interesting to see like, so the brown is the olfactory. So I guess it's good to look at the control first in this case, right?
Mm,
Because like unless you're some expert in the tissue, right? You're like, I don't even know where the brown's supposed to be.
exactly.
And it's lining this whole epithelial layer, right? It's like cells that are right at the surface and seem to extend down. I want to zoom in because I wanted to see closer. It looks like they have um, there are cell bodies both in that sort of dense area as well as something that is on the very surface.
Yes.
Um, and then presumably those cell bodies, these are olfactory neurons, so
Mm,
they have their cell body, and then they're extending, right? Some axons are inner touching them and extending back. Well, then they'll make a circuit, right, that goes back to the brain section.
yeah.
Um,
Uh, Yeah, I
Or
mean,
are there axons going? Oh no, maybe actually it's the other. It looks like the cell bodies sit inside of the layer, and then what's up there is what their tendrils are going towards, right? Because I
I
don't see any nuclei in that top surface as
yeah,
much.
I mean it's
Yeah,
weird with this though because sometimes like you can cut through a cell and not get everything because it's going in a different direction.
that's true.
So I mean,
Olfactory neurons. I'm just looking at.
yeah,
I'm seeing if we can see the. If we can see what we're looking at. Oh yeah, they look. They no. This is. I mean, that's what the schematic diagrams of them look like. The cell body sits like in that layer of cells, and then they extend like
some tendrils like
some
down.
tendrils upwards and down, both upwards and downwards.
Okay,
Yeah.
so I
Yeah.
mean, yeah, I mean it's interesting because I mean the nasal terminals are like stained completely red, but it looks at from what I'm looking at, so it's quite difficult for me to pass
Yeah,
out.
and
But
you can tell that it is those cells though, right? Because of that unique morphology, right? That we know
right,
that these cells they have that kind of hazy area of brown that exists right at the interface between presumably air and the cell, and
yeah,
then they have some cell body below, right? So when you switch back to C and now you're looking at where does the red appear? Well, you see it's like a little section of the one above, right? But it also correlates down, right? That the red goes through that layer a little bit, probably to one of the cell bodies down there. So it doesn't affect all. It doesn't like infect, but quite a few of them, almost all of them, I would say, right,
yeah.
have that antigen on them.
Yeah, and I think E, F, G, and H are interesting because like E is like the control, like uninfected, and then the rest of them are looking at. So F is infected 2 days post infection, G is
Yep.
7 days post infection, and H is 14 days post infection. And what I think they're trying showing is like the damage that is being done to these olfactory cells. Um,
Yeah, so they're not overlaying us the antigen anymore, right?
yeah,
They're just giving us the neurons. So we're just looking at how the neurons are changing over the 3 days, F, G, and H, right?
yeah,
Um, I mean, certainly G, you can tell that that surface does not look
yeah. That
does
looks
not look the same anymore. Yeah.
that definitely looks quite rough. So, I mean,
Hmm.
that's quite an interesting finding in terms of like people looking at the sense of smell, and yeah, I mean, even looking at comparisons with all
But
the rest
then
of them,
H looks a lot nicer to me again,
yeah,
right? Oh,
yeah,
so what are they when they're saying the text and they cite H? Are they saying it's healed up or they're just extended data H? Yeah, repairing.
yeah, it's uh all been. It looks like it's been repaired. Um, sorry, there's been a
So I guess they have
bit.
some longer time spans as well. But yeah, definitely like 14 days afterwards, I would say that epithelium or those olfactory neurons are back.
Yeah, yeah. So that's been and finally they look at the duodenum. Um, So looking at whether there's
The
any
first section of the small intestine.
yes, uh,
I've also
so
heard
yeah,
it pronounced duodenum, but I have
yeah,
no idea.
I think they're both correct. I
Oh,
don't think
okay.
I talked to enough people about science for it to have been settled yet. This I'm also like I think most of these words are like in people's heads before they. So it's the first time we talked to them at a conference. Everyone's like amino acid, like.
Yeah,
I
yeah.
was like, well, it seemed like that on the page, but I just don't get to talk to people about that. I mean, well, if you prefer duodenum or duodenum in the comments, just
Yeah, just let us know in the comments.
yeah, let us know. Like, comment, and subscribe. Yeah, the interesting they do actually find a focus of infection in the duodenum. They've
Hmm.
duodenum, the
So that's
which
K and L. Wait, did we
K and
talk?
L,
We didn't
yes.
talk about I and oh, that's also the nasal terminate I and J.
Yeah, I and J were the nasal turbinates. Sorry, yeah, we did talk a bit about those
Yep.
before we went in. So yeah, again, this
Yes.
shows
I know a little bit more of this morphology than I do. Well, certainly the nasal turbinate and certainly the brain, right? This is the
yeah,
villi, right?
yeah,
The villus structure where nutrients get absorbed.
yeah, the finger structure that you like get taught. Well, I got taught in school where like to absorb lots of things,
Yeah.
and
And again, that's
really,
a epithelial layer, right? Because it's like the outside of the body, technically, your
yeah,
your gut, just
and
covered in
it's
mucus and stuff.
yeah. I mean, it's interesting because I mean, when you are infecting animals nasally, I mean, sometimes there is a bit that goes down the like kind of like uh, uh
Yeah. I mean, it's the
esophagus,
same. Yeah, there's
and
two
yeah,
pipes, right? It's like the windpipe and the food pipe, so they can
yeah,
go down both ways. And even if you're sick and you're sneezing and stuff is coming up
yeah.
that way, you can still swallow some of that mucus, right? And some
Oh
of that
yeah.
can be getting into your gut.
I
Um,
mean, I think that's been like I was encouraged to do that in when I was a kid because people got sick of my runny noses, so like just swallow it, like
swallow it.
yeah, just
Oh, like to prevent like you wiping it around the classroom.
yeah, just to prevent me wiping it or using up all my tissues or just just do that, just like
Sure, sure. Yeah, so
yeah.
they
So
do this detection and are they report what it is, and they say that they see they do
uh,
see some.
yeah,
Yeah.
and yeah, we can see that we also see some. So I mean, you can't really. I mean, yeah, that's
I
so
can't.
that dems.
We can see it. Oh, the extended. Maybe it's easier in the extended
Oh,
figure.
in L, they've got like an arrow that points at the kind of dark patch of infected cells that's staying. So in figure in
Oh yeah, I
one
see
L,
it. I
yeah,
see it. I see it.
yeah, it's very subtle. They did put a yeah. Um,
That's
so
weird. That's a weird patch.
it is a
Right,
weird
like that's
patch.
not like right, like those are the villas are the ones beside, and that's just like a patch, a random patch of
That's like the base of a villus. So villi they go in all sorts of directions. So that when you
payers patch, maybe. You know that like right there are some immune there are parts of the
oh
small
yeah
intestine that have immune function. Like maybe that's what it is. I mean, right, this is the whole thing where it's like um because like I know about the surface a bit more, like there's lots of heterogeneity, right, along that
yeah
surface. And the fact that it's just in these foci, right, which aren't like, yeah, just in these foci,
yeah
it
that's a
makes
good
me
point.
wonder like what what's driving it toward localization, right? They could think of a cell type they think it's infecting and then show it. And maybe they did, but it's negative data, so they didn't want to show it to us. That's I find that frustrating, right? Because like if they were thinking it, right, and they tried it, I'd like to show it. I'd like them to show it if they didn't find anything.
I mean, there's a whole section of the extended data figures that I'm not going to be showing because they're basically looked at like the other organs such as uh,
Right.
the kidneys, the heart, the liver. They're all
Yeah.
showing signs of perfectly healthy tissue because showing
Yeah.
that oh we didn't spot anything there. So
Which
I
is,
mean that
I mean, which I think is very useful to know
yeah
because, well, first of all, I think brain is important in this model because
oh yeah
the mice that get infected die from a brain disease or like they
that
can die from a brain disease, I think is what was said.
I
There's
mean yeah.
something, yeah, sorry, go
So
ahead.
yeah, I mean the things that can happen in some infections. I know I had this with mice is if you give them an infection that's too severe, the bit of like tissue that separates the brain is called the cribriform plate, and
Hmm.
that can start getting inflammatory damage, and like but viruses and bacteria can end up flowing freely through it, and
Right,
that can cause some a short-term brain infection,
right.
uh,
And that's
which
not something that we see. We don't typically think of that as happening in humans during
mm,
this disease. So we
yeah
wouldn't want it to go. We wouldn't want to have infected these animals so much that the disease is now like not similar enough, right, to us. That
yeah
would be kind of going too far, or yeah, and it
yeah
imposes limitations on the model.
Yeah. Uh, and now I think we can go to the graphs because I think that
Yeah.
yeah
So we know. So from this, we know that they're able to cause infection in this hamster, right?
hmm
That the virus does replicate, presumably, uh, at least over the first couple days. You can find infectious virus like as early as
as like two yeah as early as day one I
yeah
think
in the lungs. You can
yep
find RNA in the bloodstream, but we don't find any infectious virus,
yep
presumably. So seems kind of like what we see in humans, right? Like that's like sort
Yeah,
of the similarity that they're going. I guess they're trying to weakly say that, it infects the olfactory neurons, just like that sense of smell loss in humans,
yeah,
right? Like they're trying to draw that parallel a little bit.
yeah,
And I think it's known that like humans can shed some right viral particles through the gut, so like
yeah.
that's why they also show the duodenum. But they don't go into too much detail on that respect. So we have this characterization, right, of a hamster that got infected. So now
Yes.
what? What are we gonna do? What are we gonna do
Hmm.
with infected hamsters, right? Um,
So and yeah, so what they did was they infected them and then then put them in cages with uninfected hamsters to see what would happen.
yeah. So for transmission, because I guess this makes sense too, because these animals can get like this is we haven't manipulated right their ACE2 at all, right? It
Hmm.
seems like this is like a lucky coincidence
Yeah.
between hamster ACE2 and our ACE2 that might be a way to study right how because receptor entry is like the first stage of viral infection, right? And they're
Hmm.
showing that virus is being shedded out of the nose. Well, those are the two things you need to study transmission, or you might need to study transmission. So yeah, turn it into a transmission model and infect one and introduce it into a cage with others uninfected.
Yes. In fact, so they did like so. I mean, they measured the weight. So when animals get infected, they tend to like stop eating, they or drinking, or and they get a bit dehydrated and their weight goes down. And then when they get better, the weight goes up. So you can sort of see that curve in like the so the first like the blue lines are like the donors, the ones that are just were infected directly by people, and then the second one down was where they took they left these donors for 1 day, and then they put them into a cage with these other non-infected mice and
Hmm.
hamsters, and then measured to see what kind of their infection was like, and it is
Yes.
kind of interesting.
Oh, I do want to say like this measurement of weight, like that's known for a lot of different animal
Mm,
models as well. Like we said, they previously already did a classic SARS-CoV infection on these hamsters,
mm,
right? And so they have these tools from that previous work, right?
yeah,
Like, yeah, they know this is what they should measure um,
yeah,
for disease. So yeah, so in C and D, those are from the hamster that was introduced into the naive cage was 1 day post-infection
yes, and then
versus E and F, where it's
6 days
later,
post.
6 days post-infection.
So yeah, so
Yeah.
yeah, this is quite interesting because it almost defines the infectious phase of for the hamsters, where they go into a cage and like they're almost like instantly infectious, where at 1 day's post inoculation
Hmm.
you can see like the recipient mice almost immediately,
Post-contact.
yeah, close contact. They literally put in the
Post-contact.
cages and left with that, and it's not there's no
Yeah.
like big delay. The recipient hamsters almost get instantly infected, whereas after 6 days the maybe only like one of them shows a little bit of evidence of having some RNA in there
Hmm.
at like
Hmm.
2 time points, and
Right.
they don't seem to get sick at all because the weights just keep going up, which is what you'd expect for like a healthy hamster. Um,
And so 6 days, they don't have the 6 days data of doing the inoculation of the nose to look at right
like
infectious viral particles, but
yeah,
presumably it's like it's between 5 and 7, so it actually could be at 6 days that they don't have any infectious virus anymore.
well, I mean, they basically they did check actually at 6 days. So if you look at the first donor graph, you can see like the tick samples at 2, 4,
Ah,
6,
yes, I
and
see.
so they
Right,
still have
so they still have viral RNA, but they don't have any infectious virus.
yeah.
Yeah,
So
because the bar graph in this case is the infectious
the
virus,
yeah.
right? Yeah. Hmm.
So again, that kind of feeds into the story about how the viral load and viral titer are different. So the viral load is the amount of RNA, and the viral titer is the infectious particles. And this
Hmm.
again puts the idea that the viral load isn't the same, so it doesn't necessarily
Yes.
show the same level of infectiousness, which is
Yes.
and which is quite, I guess, at this stage, it's quite an important thing to highlight because we're testing people for coronavirus, and so it's
Yeah.
important to be able to distinguish between live and infectious coronavirus and just that they've had coronavirus in the past and may not be infectious.
Yeah, absolutely. I mean, this is some evidence to support that idea that if you did have RNA load, it doesn't necessarily mean that you're going to be infectious.
Yeah.
Right.
So yeah,
Um, but again,
but
in
he's
like modeled in
Yeah,
an animal.
modeled. Yeah, we don't. I mean,
Mhm.
this is in hamsters, so for hamsters we'd have this advice, but for humans not yet.
And
Yeah,
we don't also know like the type of behaviors that hamsters might have in a cage together, right? Like what are they doing that that promotes the
yeah.
transfer,
I
right? Maybe they
mean,
sniff each other's noses, they might do nose touching,
from
right?
the experiments I did, I found there was a lot of variation depending on like. So if you had, so there's certain
Mm.
animals and mice. So sometimes you get animals that are dominant or who are particularly active and somewhat violent, and they tend to infect more. I
Sure.
mean, uh, so
Oh,
yeah,
were
I
you
mean,
doing transmission studies with flesh eating?
yeah, with flesh-eating disease. So
I see.
I mean, it wasn't especially successful because I feel like uh, I won't talk about it too much, but I mean,
Okay.
we did get some transmission, but the disease that transmission produced was not the same as the disease that was produced by it. So it was a very
What?
different,
Well,
less severe.
that's another important question to ask of this model, right?
Yeah,
Is the disease that's being produced here the same as uh, the one that we saw initially, right? When they showed us that histology.
yeah.
Um,
I
but
mean,
I don't believe they don't provide that for us.
yeah. I mean, that would be quite an interesting thing. But I mean, I
Right.
guess looking at the weights, the sickness kind of behaviors of sorry, the sickness kind of behaviors of the mice are probably the same. So they are get.
Yeah.
So yeah, I mean, with
Less severe, correlated with maybe less virus. Not really, actually. The viral load looks like
yeah.
it goes up almost to the same amount,
Yeah,
right?
yeah. I mean, this
uh,
is where the researchers did kind of get on my nerves a little bit because they did the whole they're not significantly that different, therefore they're significantly the same kind of fallacy
yeah,
with like
uh,
that. We got a point one on the man Whitney,
yeah.
therefore. I mean, it's
Who
a
knows?
fair
That's that. Yeah.
it's a fairly common mistake. So, and obviously doing
Well,
tests of
also, no, but because to me, this is also like, but the weight is not necessarily the disease itself, right? Like that's
yeah
like we're matching the weight to our understanding of the histology, but probably more meaningful is to see the immune infiltrate, right? Like
yeah
the amount or the date of the immune infiltrate, and the slides even don't quantify that, right? Like you don't get a
yeah
quantification.
mm,
If you want it, I mean, this is where like I think where cost always comes into play with
yes
science, right? Because I would be like, well, if you wanted to quantify this disease, right? Like they did show the CD, the infiltrates, the CD whatever for infiltrates. They could do washes and they could do cell sorting on those,
yeah
right? And they could show like immune infiltrates, and then that's like to me that's like closer to like a disease phenotype than the weight necessarily, but like
yeah
weight's easier, right? And
yeah
less work and less money to do.
yeah
Um,
yeah,
mhm.
basically, like everybody who owns animals is always doing daily weight tracking. So
Right.
if you can use weight to show anything, you do because it's just
Yep.
there. You
Yep.
have to. People who
Yep.
look after animals have to keep a track of their weights anyway to make sure that they're
Yep.
eating properly and they're keeping healthy.
Yeah. And I'm probably I have some confidence that like these investigators or other like once published, people will contact these investigators and there'll be discussion about these other things you
Oh
could measure, right? To
oh
get
yeah
get the disease. Especially if they're going to then use them all to show some effect of something, right? Like that's the other part. Like what are the interventions they may choose to add into this baseline? Uh,
exactly
Yeah, but for our baseline scenario, yeah, I'm actually impressed that the virus replicates in the animals, right? To me,
yeah
that's a good sign, right? That like even just this contact dose results in similar viral titers and loads, right,
mm,
to when they were inoculated with a drip in the nose,
yeah
right? So yeah, so C 3 days post contact. I like how similar it is to A 2 days post inoculation.
yeah
I see that as
and
very
like
promising. And then, of course, the shapes of the curves between B and D, right? I see that as very promising to indicating a similar disease.
yeah,
Mm.
and even and the viral loads seem like I mean there is some like kind of more variation in them, but then
Mhm.
you'd sort of expect that because of a natural transmission.
Yeah,
And
exactly. They're not
yeah,
getting the precise micropipetted in liquid.
yeah, I mean it'd be very interesting to see if there's like gut like diseases in the transmitted animals because then you can say that for sure that okay some this is something that will naturally happen.
Mm.
Um, uh, and so I think we can move on to the next slides, which
Yeah, I
I
think
think
so.
Okay, so this one was where they did so I'm showing up the cute little diagram they did of the mice in the cages, demonstrating the aerosol contact,
Yep.
and the hamsters violently disagreeing with the 6 foot rule, breaking it, and it's a 1.8 centimeter rule.
1.8 centimeters. Which, I mean, you have to
Naughty, naughty.
you have to scale it to their bodies, though, right? Like,
Yeah, that's true.
We're a lot
But-
bigger than hamsters, but I
True.
still think that's going to be under their whatever it is
Yeah.
they need to
And
maintain.
So yeah, it's interesting because they actually took these. They put these mice into these like these cages within this like bigger like air circulator,
Mhm.
and they basically left them there for about 8 hours, and then they took them away and put them into their separate cages to check whether transmission had happened,
Mhm.
and we're going to see the results of that. So this is on figure 3. So figure 3A, B, and C shows like the donor
Yep.
mice,
Mhm.
and the ones that we're
The
curious
donor
about
hamsters. You said
donor
mice like for the
hamsters.
last 3 times.
Yeah, that's
That's
this is
fine.
why I warned people. I was gonna do that. I'm gonna do that. Okay, right. So, and in D, E, and F, we see the recipient humans getting the I did it again.
Yeah.
Yeah, but this is
yeah.
quite interesting because like they were actually exposed for a very short time, no physical contact, and only through air, and you can see that they too get an infection that looks yeah.
Yeah, and I mean, I think what's cool for me in this is that so now they're restricting the physical space, right? Like the first in Figure 2, the hamsters are doing whatever they want, right?
Yeah.
Roaming around that space. But then in Figure 3, they're adding this new restriction on the hamsters. But you see a very similar profile, I would say.
Yeah.
Yeah. So these 2-day. Are these 2-day post-infected as well? The same.
Yeah. So I think they were also I think they were mixed in at like 2 days post-infection.
Yeah.
Yeah. Also, they were exposed to the donor animals on one day post-infection for about 8 hours.
Yeah. Yeah, 8 hours in a room with another hamster.
Yeah, I know. And they didn't even have to watch 2 Lord of the Rings movies. I mean, it's
Yeah, what do they get to do? What their enrichment was to look at each other.
Yeah, I guess so. Because
they got some social contact. They can squeak.
True. And the interesting thing they also do here is they look at fomites. So they took like a cage that had been filled with infected hamsters. They took those hamsters out and put in naive hamsters to check whether they would get infected.
Mm-hmm.
And this is actually quite interesting because they like see that like one of those. So there's n number of 3, so quite low, but and it seems only 1 out of 3 tended to get the infection.
Mm-hmm.
From the fomites, but
I mean, this is the only experiments they presumably did, right? So it's like,
yeah.
In this experiment, 1 got the infection.
Yeah, but I mean, it's interesting because they've got like an extended data table 1 where they look at the different donor cages that they gave and they swab them to see whether those cages had some RNA or live virus around.
Well, they swabbed all the places in the cage, right? Like
yeah. So sample area, bedding, material, corn cobs.
Yeah.
Cage side, plastic, pre-filter, paper-based. And of those, it seemed that only 1 out of 3 cages seemed to have live virus in it, by the looks of it.
And it was on the water bottle nozzle. Oh, and the cage side, both hard surfaces.
Yeah.
But those could also be easier to sample. Like, I think it's hard to. Well, I mean, the filter is probably easy to sample, but the bedding sounds super hard to sample. How are you going to sample bedding and say that you found virus on it?
It's you have to like kind of shake it. Like I guess you have those
ones like which bedding? Yeah. Like there's so much of it. Anyways.
Yeah, I mean it's interesting because like since it's at day 4, it's that gets difficult to pass out whether the animal got it from the water bottle or whether they put it on the water bottle. So
afterwards, that's right.
After,
that's right. So, it's a bit early, and they don't have all those things on day 2. They didn't do all the swabs on day 2.
I mean, I understand that because I mean, luckily we got the reviews of this paper. So the Nature have put in the supplementary material, the reviews online,
Hmm.
and these like kind of fomites experiments were pretty much asked for by the reviewers, so
Yep.
they're added at the last minute. So
Yep.
what I imagine happened was that the authors were like, okay, we'll swap this, and then as they go, like, wait a minute, should we do more? We should
Yeah,
probably do
yeah, yeah,
more.
yeah,
We
yeah,
should do
exactly.
more.
I imagine the same thing then, yeah, because it was like they set it up and then they're like, okay, we did it. We're gonna get this data in, and they're like, well, we're doing it. We might as well do all this other stuff. Yeah.
Yeah.
Which again, it's
Yeah,
good because it tells a story, right? It helps us tell the story, and this is the thing about the whole science.
hmm,
Even we touched on this, like when we're talking about like even developing the model in the first place. Like we're only going to understand what people record and tell us.
yeah,
All those other things are like the scientist who does the experiment understands, right? They've done it,
hmm,
so like they have that experience, but like they're just one person, and maybe multiple minds have to come together to try to figure out what the pattern is, or get in a new idea for another direction. And so yeah, so it's really great that they did end up collecting this data. But yeah, I
yeah.
think the data has a hole in it that makes it not as useful as it could have been.
Yeah, I mean, because again, I'd say that the fomites data is kind of inconclusive because because I mean, if I were doing it, I'd have like dunked a water bottle in the in the virus and then like at
Mm.
different doses and then
Yeah.
tried to check it out.
Yeah, well, I
But
mean, that's sort of what's suggested by their study, right? Like, it's like if you want to go on the hunch, right, that oh, this is how it might work, right? That interpretation of seeing the water bottle have it, then like, yeah, let's put some in the cage in different areas and see
yeah,
how the hamsters get it, because we know the hamsters get it from just being in the same room as each other. What about being
yeah.
in the room with an object that has it on it?
Oh yeah. I mean, I remember like thinking about trying to buy a nebulizer for my transmission experiments, but
Mm,
like they can be quite like expensive and they're not necessarily like as representative as you'd like. So that's
right,
why that using like a proper animal to do it is a
right.
bit better than that.
Yeah.
But yeah, I
And
think
so
that
like I think
yeah,
what's I will say that when we were reading those reviews that they had posted, they put a lot of emphasis on the fact that paper had come out already about this Syrian hamster model, right?
yes,
And you can find that. I think that's reference 24 or 27. Yeah,
yeah,
reference 27 in this paper is um, yeah, is COVID-19 and the golden Syrian hamster model. So another group had already done these experiments. Um,
yes,
and in that
uh,
one, they actually looked at the intervention, like sort of the relevance. So they have the same setup. Well, first of all, they use like both male and female hamsters, I think, in their setup. But again, they're doing just nose infections. And yeah, they're measuring the same stuff. They're measuring plaque assays and RNA values, and I think all of that is similar to
yeah.
what they're reporting here. But the intervention they look at is convalescent serum, prophylactic convalescent serum, which is interesting. You
Yeah.
know, like because like that's almost treating it more like an infection model
Well,
in
yeah.
that sense.
I
Yeah.
mean, yeah, I think there's an argument to be had for like just pure characterization papers without having that treatment in mind. Like, so
Mhm.
this
Mhm.
because I mean, I saw a similar paper on golden hamsters where they were testing face masks, where
Yes.
they had face mask material in between like some air circulated between hamster cages, but with that you've lost some of the other aspects of like fomite contamination and like kind of another thing that like I would have liked to see,
Well,
but
I don't even think they're losing it. I think it's a different
yeah,
focus, right?
exactly,
It's
you're right.
like the core of the core of what I think excited us to see this paper, or I guess I'll speak for myself, but the core of what excited me about this paper is seeing like here is here's an instance, right, where we have a human virus, we're putting in an animal, and it happens to transmit from that animal to another animal of the same kind, right? And that seems like a really useful just setup,
Mm,
right? That you can imagine all these different things that you could do with that setup that would tell you little tidbits of information, right? That might be suggestive one way or another.
yeah.
Um, and that's the model, right? But then when they string that into a story, right? Like you want to say like, but what does the model tell you? And you could go in so many different directions. In this paper, they chose to move the animals between cages, or like, right, restrict the physical
Yeah.
space of the animals to speak about something about transmissibility and like where the virus might be coming from, right, in
Yeah.
the transmission cycle. But we saw that they do have some sort of lung pathology,
Yeah.
so you could go down the route of some sort of disease model as well, right?
Yeah.
And give them serum and see if they still get disease.
I mean, there's
And
also the whole olfactory story that they did delve into a bit more than that. I mean, that was quite a nice
yeah,
surprise for me. And
yeah,
again, that's something that could, because I mean, because again, you can't really tell whether an animal can't smell anything without doing some clever like psychological experiments. So
sure.
so actually, like this
Well,
is
actually, I mean, this is going to leverage knowledge of looking at that graphical abstract last week of the mouse paper. But
mm,
in the mouse model, they used adenovirus, right, to
right.
deliver the H2 receptor onto the lungs.
Yeah.
Well, does that might not mean like that might be enough for the virus to get in and cause infection, but maybe that doesn't infect the neurons
Yeah. Exactly.
in the turbinates. And so like in that mouse model, like you're missing out because they focus so hard on just getting the virus to replicate in the lungs that you can't see some of the other stuff, right? Where animal that natively has the right protein
Yeah.
or a protein that's amenable to that infection, you can study that stuff because like you don't have to set it up. Biology did it for you already.
Yeah.
Right? They already have a distribution of expression in their bodies.
Yeah, and I do like that they did try to look at all the other tissues as well to see where else the coronavirus could disseminate to. Because I mean,
Mm-hmm.
admittedly, it didn't pay off for like a lot of the tissue they looked at, but where it does pay off quite well for them. The spotting,
Mm-hmm.
it's a good spot to see it in the duodenum. I mean, yeah, potentially the Peyer's patch. I mean, those are quite. I mean,
I'm also thinking that what distinguish like the other reason why I would say that it seems more transmission model-y to me is that if they were going for an infectious model, I think one of the things about infection is really useful to know is also like severity, dose response kind of deal. They don't do
yes.
that, right? They don't show us that like with small amounts they get this type of disease, with large amounts
Mm,
they get this type. So like that does like make it more limited on the pathogenesis. Not to say that they couldn't do those studies, right?
yeah,
And you could move forward with a hamster as a pathogenesis or like as a disease model. But yeah, as a transmission model, I like it. I like that they don't have to do all these manipulations of the animal.
yeah,
Yeah, and like to make them susceptible. Like they're already previously susceptible, um, which is nice.
yeah. Yeah, I think that's. I think I've got covered all the figures in the paper. So let me check. Yeah, no, I've run through the list of figures, and yeah, and we
Yeah.
think we talked a bit about like how relevant this is going to be. Um,
Yeah, well, another thing that I pulled, I guess I do want to say a little bit more about relevance, right? Because like, what is the model? So it seems like they've used the model three ways now, right? Or
mm,
like in broad strokes, right?
right.
Syrian hamsters. They've used it in three ways. They've shown us something. Maybe you can tell us something about face masks,
Yep.
right? By using basically replacing filters with face masks,
Yes.
right? And saying that like, do face masks block right the air? And like, you can imagine a limitation of that is that like, well, those face masks aren't made for the types of aerosol particles that hamsters make, right? Do
Yeah.
hamsters make the same aerosolized particles as we do? And also like they're working at such different scales,
Mm,
right? Like right, their close contact is this 1.8 centimeters, right? That's not really our close contact,
yeah.
right? And the physics of those particles, I don't understand, right? So
Yeah.
that's a weird one. I think that's a strange direction to go. I can see why people might want to go that direction,
I mean,
but I'm not sure. Like there's always that doubt, right? Like how useful is it going to be? And it just takes a scientist who's convinced of the usefulness of the data to end up making those papers.
yeah. I mean, I know that there were like certain studies were done in the 60s with like strep where they actually put petri dishes at certain distances from people to see the range of how far they could
Ah,
spread. So they had this
yeah.
nurse like do jumping jacks who was like infected with like the disease and put these petri dishes at certain distances to figure out how far the like just her doing lots of exercise could get the. I mean, maybe doing something like
Interesting.
that for coronavirus. I mean, in a
I
more
mean,
ethical
they
sense.
had right, but in that sense, the strep that they were transmitting that didn't cause super serious disease, right?
It was
Or
the.
did
I
it?
mean, thing about strep is strep pyogenes. It both causes a sore throat and the flesh-eating disease, depending on where it lands on you. So,
Right. Right.
most of the
So,
time,
but that's the thing. It's like
yeah,
that type of exposure, it wasn't going to expose you to like a high risk event.
yeah,
Like you'd need some sort of like changed virus, right? Like maybe if they had the attenuated strain of the virus, right? If like let's say we have our
yeah.
vaccine strain of the virus, and the vaccine strain is known to only differ in this ability, like to replicate inside of cells. Well, then that virus would be great for these types of studies, right?
Yeah.
Because like you could give it to humans and it wouldn't be a big deal, and you could start looking at these types of dynamics.
I mean, yeah, I mean, I guess there might be studies already out about just droplets and like how well viruses get, because
Yeah,
actually,
those are the studies I've seen in masks. Like
yeah,
they've done mask studies where they have humans blow through masks and put it on plates, right? Like
yeah,
almost like that almost sounds like a better model to me than the hamster style of doing it.
because yeah, with face masks and respiratory, it's almost like physics at that point. You need someone to think about like kind of the flow rate, like
Hmm.
the kind of droplet size and all the physics associated with
Right.
that.
Right.
But
So then, okay, so like, let's see. So then they tried convalescent serum, right? So
yeah,
this idea of like, so this is like a therapeutic. So it's like, oh, they're really speaking about, in some ways, they're really speaking about the dose that gets transferred at these contact events,
mm,
right? That's sort of what they're studying at this point. They're
yeah,
saying like, when you get a dose that's comparable to, I don't know, how animals transmit it between each other, right? Can the intervention of giving you a few antibodies, right, be successful in prohibiting the spread?
mm,
I think I think it's interesting. I mean, I would actually think, to me, it's more interesting to see what's on the shedder's side, right? Is there
mm,
something that you could change on the infected animals
yeah,
that makes them less likely to transmit to us, right? Mechanism, like, yeah, and dive into that mechanism. I want to see. To me, this model is useful from the shedding perspective.
yeah,
Um, mhm. And
uh,
the interventions that come from that, right? Because like showing interventions on this in this model and the outcome is how many people you infect, I think that's really, that's of public health concern. That's
yeah,
useful stuff. And that's where you could see some therapeutics going, I guess. So this model may have utility in that respect.
mm, yeah.
Uh,
So I
yeah. And then the olfactory stuff, right? But they don't really follow up on that. But, this is just the report of what they found.
yeah,
We don't know
this is
what
like
will come of it.
yeah, I mean, this all been done so fast, and we are like learning so much about this virus as it's happening. And I
Hmm.
think like we're drawing to the end of our time.
Yeah,
So
absolutely.
yeah, thank you so much for joining me. This has been excellent to share this paper with you and talk and discuss it.
Yeah, absolutely. I think it's been really great. And next week we'll be diving back into the stack of papers that are getting published every day on this subject.
Yeah,
Trying to find something interesting to talk about.
yeah, digging for gold to
Yeah.
find the papers are the most
So,
yeah.
so if
So
you have anything interesting that you would like us to consider, please tweet us at either our handles below or the hashtag Micro Twitter Journal Club.
yeah, and also like, comment, subscribe because I've been told that that's what you should do when you're in YouTube. uh, Ding the bell.
Oh yeah.
Stalk us. I don't stalk us.
Stalk
I
us.
don't know. I'm new to YouTube. I don't know. But anyway, thank you so much for joining us for this
Stalk him.
hour.
Just joking.
And we'll hopefully see you next week.
Yes.